This is part one of Chapter 17 on the central dogma and the process of transcribing and translation. So, first of all, we start with what does a gene code for? So, as you know, DNA, the series of nucleotides, actually are the instructions for the synthesis and production of a protein. And then those proteins, particularly in the form of enzymes, but those proteins go on to then make everything else that's actually a part of who we are and how we function. So this is how the the DNA in our cells actually encodes our life, our functioning, and who we are. The central dogma is a concept you should be familiar with. It's summarized in simply the flow of genetic information in a cell. So this flow of information from the DNA to the production of a protein, which then will display some sort of trait, which again, as we've just gone through genetics, that trait is something that defines what we see and how we function, um, how an organism looks, and just everything we are. Gene expression is defined as the process by which DNA directs the synthesis of proteins and includes two stages, the transcription and translation. So how we got to this point really was by looking at things from the malfunctioning view to actually understanding how it functions normally. So, in noticing that metabolic diseases were inherited, there was the conclusion, or suggestion at least, that genes were responsible for coding for the enzymes that were missing in these metabolic diseases. Each disease, which we could look at as a phenotype, is caused by something not working that should be made by the DNA. In most cases, this is a lack of an enzyme. So, for example, if we look at Tay-Sachs, what we're missing is the beta hexoaminidase A, which is responsible for breaking down, didn't mean to do that, is responsible for breaking down fatty acids in the cell. And so, as you know from our last unit, Tay-Sachs results, and all the symptoms of Tay-Sachs results from a buildup of fatty tissue in the body. PKU is short for phenylketonuria, and what's involved in that disorder is a enzyme, phenylalanine hydroxylase, which does not get made. And the responsibility for that enzyme is the breakdown of phenylalanine, which is an amino acid, but the buildup of that amino acid in the cells actually, particularly in neurons in the nervous tissue of the brain, the buildup of phenylalanine actually destroys the brain and causes mental retardation. And then the last example that's listed here is albinism, which results from tyrosine not being produced, and that enzyme is actually used in the production of cellular pigment. So each of those disorders actually is the result of one single enzyme not being made. And so from that, there was this idea that if these conditions are inherited, they're coming from the parent's DNA and being given to their children in that way. So trying to advance here. So Beadle and Tatum, two scientists who are famous for this one gene, one enzyme hypothesis. And how they came to that conclusion or to that hypothesis was a result of this large study that they did. They exposed bread mold to X-rays, and by doing that, they found that they were able to create 3 different mutants that were unable to survive on minimal media. And that media obviously had to do with arginine being needed. and was missing. So they used crosses to identify these three classes of arginine deficient mutants. And what they found that each of these mutants was lacking a different enzyme that was necessary for the synthesis of arginine. So what they actually discovered was that, first of all, there was a metabolic pathway involved in the production of arginine. It wasn't just a simple one-step process. But what they were able to conclude is that one gene must be involved in the production of each enzyme. This is another depiction of this experiment showing how the minimal medium was used for all four of these here, this being the wild being our control group. And what they found was when this enzyme was missing, then arginine was not produced. When this enzyme was missing, the arginine was not produced. And then when this enzyme was missing, arginine was also not produced. And what they found is there were different genes. There was gene A, B, and C that was mutated, and that resulted in this loss of production of that enzyme. So again, that one enzyme, or one gene, one enzyme, or one protein idea came about through this study. Later, though, we have had to rethink this idea because some proteins are not enzymes, as you know, and many proteins are composed of many polypeptides. So a single gene just making a single protein is really a little simplistic, because many times there are several genes that go about making a very complex level 4 formatted gene or a protein. So now we restate Beadle and Tatum's hypothesis as one gene makes one polypeptide, and then there can be several polypeptides that go together to actually make a protein or an enzyme. So transcription, I love this picture of someone writing here, because transcribing means to write something down. And transcription to me is changing DNA's nucleic acid language and writing it into RNA's nucleic acid language. I think that's a great way to remember how DNA gets written into RNA. And if we look at RNA, this is, I'm sure, a review for you, but some of the differences between RNA and DNA are that RNA is made from ribose instead of deoxyribose as its sugar. In terms of its nitrogenous bases, it uses uracil instead of thymine, but other than that, it has the other three, the adenine, guanine, and cytosine. The base pairing is the uracil will base pair with the adenine. Cytosine, again, base pairs with guanine. Another big difference is that RNA is single-stranded as composed to DNA. And a last but large difference between RNA and DNA is that there are a lot of different types of RNA in a single eukaryotic cell. There are several types in prokaryotes as well, but not as many as eukaryotes. They actually are discovering more and more, and they're simply calling a lot of them just NC RNAs because for non-coding, because they're still trying to figure out what exactly they are functioning as and what they do. And it's still a process of discovery when it comes to these different RNAs. But we'll define 4 types of RNA here. Messenger RNA, as you know, is the one that encodes the amino acid sequence that is going to be translated into a polypeptide. It's the one that is actually read from and produced from the DNA. Transfer RNA brings amino acids to ribosomes during translation. Ribosomal RNA makes up ribosomal proteins that make up the ribosomes, along with some other proteins. And as you know, ribosomes are the organelles that translate the messenger RNA into protein. Then the SN RNAs are small nuclear RNAs, and they, together with some other proteins, form complexes that are used in RNA processing in eukaryotes. And we'll look at these a little bit later in this lecture. But it's important to note that the small nuclear RNAs are not found in prokaryotes, so you will not see them in bacteria. So first of all, we'll start with transcription. Transcription is the making of messenger RNA, and what we have is the transcribing of one DNA strand. So as you know, we have our double-stranded DNA, and when we talked about replication, we saw that replication occurs simultaneously on both strands of the DNA. But in transcription, it's very different because there's only one strand that ever gets read, and that's called the template strand. And it's something that is always consistent. It's the same strand that serves as the template. So the template strand is the one that actually you can see the messenger RNA being built on. And that building of messenger RNA is similar to replication, where it's being built 5' to 3'. So we're adding again our nucleotides to the 3' end of our growing strand. You will find what we call the coding strand is the one that is not being read. But what's important and why it's called the coding strand is because it actually gives you a preview of what your messenger RNA is going to look like. So because it's complementary to the template strand, it's also going to look like the complementary messenger RNA strand, with the only exception of the T's being replaced by U's. So I'm sorry I don't have an arrow, I just have this cross that you see. But you can look at this carefully and notice the T's and the U's. And if you look here, there's a G for the guanine, there's a G here on my messenger RNA, C, C, T, U, A, A. So this, again, shows you what your messenger RNA should look like. And this is called the coding strand. The enzyme that we use for this is RNA polymerase. And there are a number of types of RNA polymerase. And they're actually defined by what they transcribe, which genes they transcribe. So RNA polymerase 1 is the one that only transcribes the ribosomal RNA genes. So it is only responsible for getting the instructions to make ribosomes. The RNA polymerase 2 is the one that we typically think of when we're talking about transcription, because it actually is responsible for carrying the instructions or making the messenger RNA that is going to be for every other gene besides these other polymerase 1 and 3s. So basically they're getting all the DNA's instruction. So RNA polymerase 2 transcribes all genes into messenger RNA except for the ribosomal RNA gene and then the transfer RNA gene. So the transfer RNA gene is transcribed only by RNA polymerase 3. And the way these enzymes, these RNA polymerases, know which genes to transcribe is that they will only bind to a specific promoter sequence for the gene that they are responsible for transcribing. So RNA polymerase 1 is going to look for the promoter sequence for ribosomal RNase genes. And RNA polymerase 3 is going to look for the promoter sequence for the transfer RNA genes. And then RNA polymerase 2 is not quite as picky. You know, it's going to actually bind with the promoter sequence for all the other genes. Another thing about these RNA polymerases to note before we move on is that they actually are responsible for prying apart the DNA strand. So they're a much more complicated enzyme that serves the purpose of, for example, the helicase in replication that we saw. So the unwinding and unzipping that occurs in transcription is actually accomplished by the RNA polymerase. Also, RNA polymerase does not need a primer, but it does follow the same base pairing rules as DNA polymerase does in in replication. This is a picture that will show you the stages of transcription. So we have 3 stages of transcription to remember. We have stage 1, which is initiation, and that's when the RNA polymerase is going to bind to the promoter region and start transcribing. And again, remember, there's no primer needed, so it can just bind right to the DNA to start transcription. And you can see here, this is our double-stranded DNA. The RNA polymerase is able to unwind it and separate it out to begin the messenger RNA. So that's initiation. Next, we're going to have elongation. So this is now when we're going to start building our RNA transcript. So here's the RNA transcript in the red here being produced as the RNA polymerase moves down the template strand. And again, you can see the direction of transcription is towards the 5' end of the template strand. Our last step is termination of transcription, and this occurs when we have some sort of termination sequence here. And it depends on whether this is eukaryotic or prokaryotic on how that occurs. But what is common for all is that at this point, RNA polymerase is removed from the DNA, or removes itself from the DNA, and the messenger RNA is separated from the RNA polymerase as well. And again, we're calling this an RNA transcript at this point for a specific reason that we're going to look at in just a bit. Here's a short animation to show you these different steps and summarize them.
The first step of protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Okay, we'll stop there for this part 1 video.